My name is Hollie Allison, and earlier this year I obtained my PhD in biomedical engineering at NUI Galway under the supervision of Professor Leisha McNamara. Today I'm going to talk about my thesis titled Mechanobiological Changes During Estrogen Deficiency Leading to Bone Loss. So osteoporosis predominantly occurs in females, and it's been estimated that over 200 million women worldwide are affected by the disease. Osteoporosis is characterised by severe bone loss, which leads to fractures, pain and immobility. Common treatments for osteoporosis target bone loss by inhibiting osteoclast activity, but these treatments have been shown to only prevent osteoporotic fractures in 50% of sufferers. Therefore, an advanced understanding of the underlying mechanisms and treatments for osteoporosis is needed. So estrogen deficiency has been shown to alter the survival and function of osteoclasts, leading to bone loss. And this bone loss has been shown to alter the mechanical environment of the cells. Mechanically loaded osteocytes and osteoblasts have been shown to downregulate rank ligand and upregulate OPG. And when these cells are co-cultured with monocytes, a decrease in osteoclastogenesis has been observed. Estrogen deficiency, which occurs in postmenopausal osteoporosis, alters how osteocytes and osteoblasts sense and respond to mechanical loading. How these changes in osteoblasts and osteocyte mechanobiology affect osteoclastogenesis is not fully understood. So in my PhD research, I conducted a series of conditioned media and co-culture studies. And what I found was that under estrogen deficient conditions, mechanically stimulated osteoblasts failed to inhibit osteoclastogenesis, and a significant increase in osteoclast differentiation and matrix degradation was observed compared to estrogen-treated groups. I also assessed the effect of inhibiting the Rho-ROCK pathway on osteoblast-induced osteoclastogenesis. And what I found was that when ROCK was inhibited in osteoblasts, this exacerbated osteoblast-induced osteoclastogenesis under estrogen-deficient conditions. So from these studies, I proposed that a reduction in OPG expression by estrogen-deficient osteoblasts in response to mechanical loading may leave osteoclast differentiation and matrix activity unchecked. So in addition to those studies, I also assessed the effect of estrogen deficiency and mechanical loading on osteocyte-induced osteoclastogenesis. And similarly, what I found was that under estrogen-deficient conditions, an increase in the rank L to OPG ratio expression was observed in estrogen-deficient osteocytes. And when co-cultured with bone marrow macrophage cells, this led to an increase in osteoclast differentiation and bone resorption. I also showed that inhibiting sclerostin by use of a sclerostin antibody reduced pro-osteoclastogenic signaling between osteocytes and osteoclasts, and this led to a reduction in osteoclastogenesis and a reduction in bone resorption. So in addition to these in vitro cell culture experiments, I also conducted ex vivo experiments looking at the temporal changes that occur in cortical microporosity during estrogen deficiency using an overoptimized rat model. I found that increases in vascular canal diameter seen in short-term estrogen deficiency were reverted in long-term estrogen deficiency. In addition, in long-term estrogen deficient animals, there was a decrease in lacuna porosity and lacuna diameter, as well as an increased incidence in the amount of empty lacunae. These temporal changes that occur during estrogen deficiency may have implications on the mechanical strains experienced by osteocytes, leading to secondary mineralization changes. So the findings of my thesis highlights the importance of effective mechanical loading for the inhibition of osteoclastogenesis, thus indicating that future treatments for postmenopausal osteoporosis should aim to restore loss of mechanosensitivity during estrogen deficiency to revert bone loss.